Welcome back, everyone. So now we're ready to conduct the actual procedure. I'd like to explain how it's going to work for your report. You will have to work a little faster on it because your TAs have to have enough time to grade your work, and I have to have your final grades figured out. So your lab is on Tuesdays and Wednesdays. And in a typical year, in a typical situation, you would have completed this report right here in lab. So you wouldn't have a whole week to do it. So please plan to spend a little bit of extra time. After watching these videos, just go ahead and complete the entire report. And you will have until Sunday night to complete it that same week. You will not have until the next Tuesday or Wednesday like you typically would, because again, TAs need to complete the work. But if you follow my directions, and we will discuss how these data should be filled out in the report sheet, you should be able to complete it fairly quickly. So let's talk about preparation of the setup first. So this is a reaction flask. This is where the tablet will be. In your procedure that hopefully you've looked over, you're supposed to take a piece of tablet and hang it on a piece of thread. I decided to do it slightly differently. I'm gonna drop the tablet where acid will already be sitting, and I'll quickly stopper the flask with the stopper over here. But that's the last thing we're gonna do to actually start the experiment. First, we have to prepare our setup. And one thing I wanted to show you is the leveling bulb. Again, if you look at the directions, we're supposed to add some water to the leveling bulb. But it's not just water. It's actually water with about 2 milliliters of hydrochloric acid, 6 molar HCl, and tablet crumbs. So what I'm going to do is I'm going to take another tablet from this packet. I'm going to put it on a piece of paper towel. I'm trying to handle it as little as possible with my bare hands, because moisture from my hands can sort of break it down. I'm going to break the tablet into pieces. So you can see it's now broken up into pieces. Maybe a little bit more. And I'm going to take a small piece here and drop it in the water. You can see it's bubbling, which we already saw it do. And maybe another small piece here. And I'm going to also add the HCl. I already pre-measured close to 2 milliliters of HCl. So why are we doing this? Why are we adding the pieces of tablet and some acid to the liquid that's ultimately going to fill our burette, the tubing, and portion of the leveling bulb? Well, this is very, very important step. We don't want just to have water in here, because if we only have pure water and CO2 happens to form in our flask and comes in from the flask into the burette, it will come in contact with the water, and guess what? It will start dissolving in it, right? Any gas dissolves in water, and we will be losing CO2, and we will have no idea how much CO2 has dissolved and lost. But by putting acid and crumbs of tablet to this liquid, we're saturating this liquid with CO2. You can see hopefully maybe tiny bubbles of it still forming. I'm going to stir this a little bit. So now this liquid has already plenty of CO2 within it. So when CO2 in our reaction comes in into the burette, it will not dissolve in this liquid, so we will be certain to measure all CO2 that has formed. So I'm going to go ahead and I'm going to add this liquid to my leveling bulb here. There are a little bit of air bubbles, so I'm trying to squeeze them out. As you can see, it probably is not enough. So I'm going to add a little bit more water. This time it can be just regular water. It can be tap water also, because we don't really need it to be very pure. Why do I need more water? Because number one, I want to be able to see the level of liquid here in the bulb. I don't want the liquid to be covered, or the top of the liquid to be covered with the ring. And now what I can do is lower the bulb a little bit to allow liquid to enter the burette. Will also help if I unstop it here. There. Now watch what happens as I move the bulb. I move the bulb. Hopefully you can see the liquid in the burette sort of following the bulb. If I move the bulb lower, liquid follows lower. If I put the bulb higher, liquid follows higher. Why is that? Well, that's the pressure we were talking about. So the pressure exerted on the liquid in the bulb right here, because the bulb is open to the surroundings, is atmospheric pressure, right? And so pressure in the burette will follow the bulb, and this is reflected in the level of liquid. So now what we need to do is we need to bring the bulb higher. I'm going to attach it back to the ring stand. Now I need to take my reaction flask and to this reaction flask I'm going to add 5 milliliters of 6 molar HCl. That's my reaction acid. And then I'm going to weigh a piece of tablet for my reaction. Now the mass of the piece is important. Again, if you read the directions will tell you that the mass of the piece you should use for a trial should be somewhere between 0.2 and 0.3 grams. I usually suggest to students that they use a mass closer to 0.2 grams rather than 0.3, just because burette is only a 50 milliliter burette, right? So if we have too big of a piece, too much CO2 will form, and we're risking the liquid basically falling down below 50, so we can't read the final volume. So because of that, I'm going to get a piece closer to 0.2 grams. And then I'm going to add my acid to the flask. I'm going to clamp the flask right here. I will then stopper the flask. We will take initial reading of the burette's volume, and then we'll continue with our experiment. So in the next video, I'm going to demonstrate all that.